guys, welcome to week 4 PowerPoint video number 2. So last PowerPoint we talked all about prokaryotes. This one is all about eukaryotic structures and organelles. And I will use this as the first opportunity to point out that you can have this information, that this video might not be the best use of your time, because all I'm going to do is go through this various PowerPoint where you might find this information more condensed in the various other videos. But I know some people really like to have me go through it. So again, I'm assuming you've already looked through this PowerPoint, and I'm not going to be reviewing every single slide. But I will talk about this slide, which is just to point out that we didn't know about cells until we had the technology to see cells. So this is one of the first places that we see the studying of biology is directly linked to the technology we have at the present, or at the present time of that. So when did we first start knowing about cells? Not until the 1600s, when the first microscopes were invented and the first use of lenses to magnify samples of interests. As the 1600s continue, the microscopes got better, involving 2 lenses. Robert Hooke, he's the one that that made the term cell for describing what these little boxes or structures that he was looking at. Your textbook goes a lot more in depth into different microscopes, and again in the PowerPoint slides that you're going to be working on. For here, I'm just going to leave it as different microscopes are needed to see different levels of resolution. Obviously, some things you can see with just your eye. I can see a person or a chicken egg. Some things you can see in a light microscope. In class, if we were in person, you would be using light microscopes to see animal and plant cells, and you'd be able to see a little bit of their underlying structures of these organelles, like the nucleus. If you were in a much fancier lab, you could use an electron microscope to see those organelles and the macromolecule levels. Some levels, at like the atomic level, you can't see with a microscope. You need to use other tools and other technologies. So here we can see, or this is the beginning of the rest of this presentation, where we're going to be starting to talk about these differences that make a eukaryotic cell a eukaryotic cell. So you already covered this information in that other PowerPoint, going through the structures of a prokaryotic or a simpler cell. So you already know these things about prokaryotic cells being smaller, not having organelles, having their DNA in the nucleoid region, not in a membrane-bound organelle. Prokaryotic cells are bacteria and archaea, and they were the first cells to evolve. So even though we haven't yet covered these, you should already actually know this information, along with what's similar to all living cells. All of them have a plasma membrane, they have cytosol, the liquid in between, they're made up of DNA with chromosomes, and then they have ribosomes that are making proteins. So again, by the end of this presentation, or by the end of your studying this unit, you need to be able to go through and be able to recognize all of these structures. For this unit, I'm going to expect you to be able to recognize things by a picture or by the function, or go reverse. If I give you a picture, can you tell me the function? If I tell you the function, can you point to where that would be in a picture? So here we have a great slide overview for an animal cell, and here we have the plant cell. So this is ultimately the level. Again, you have some great videos. We have some great videos, which I will just play for the sake of you seeing them. I don't think you can hear the sound that's going on, but the point of this is it's a great way to actually practice, is to mute and then see if you can be explaining what all these structures are. So I'll leave you those. I'm not going to go through any of these videos. And additionally, this is the type of material. Ask yourself, quiz yourself. Do you know what the answer to all of these are? I actually put them in here, so I'm not going to take the time to read them out loud to you, other than to mention, of course, that you have those similarities. Everything has DNA. Everything has that plasma membrane, but the whole definition of a eukaryotic cell is it has that membrane-bound organelles, that nucleus. So the first thing we're just going to comment on is the plasma membrane that is common in all living organisms. The entire chapter of next week is going to be devoted to talking about the plasma membrane, so I'm not going to belabor it other than to say that it's the selective barrier. It's like the bouncer at the cellular nightclub deciding what comes in and what comes out. So you do already have been introduced to the fact that it's made up of phospholipids with a hydrophilic head and hydrophobic tails made up of those fatty acids. Again, you always want to be connecting to the information we've previously learned. Okay, so now we're getting into new information. We're going to start structures of eukaryotic cell, the nucleus. You couldn't have an argument with someone that the nucleus is what's the most important because it is holding all of the DNA, which is, as we know, the genetic material that tells everything else how and what should be done. Now, when you can see this structure, you see hopefully a whole bunch of things. One, you see that there's this nuclear envelope that's made up of two layers of a bilipid membrane. We can also see that it is continuous, that membrane, with these other structures which we're going to learn about in the rough ER and the smooth ER. Hopefully you can also see these holes. So I'll talk a little bit more about those in a second. When I say the nucleus is super important because it's holding the DNA, it's storing the DNA, but it's wrapped up in specific structures called chromosomes. And we're going to talk more about this in a few more chapters, but understand now and start to have this picture that there is a ton of DNA in every cell. In fact, there's about 6 feet of DNA in every single cell. So if you think about a cell being the size of a period at the end of a sentence in a textbook, inside the nucleus of that period would be 6 feet of DNA. How do you get so much in? Because it's wrapped into these really specific structures. So the protein gets wrapped around histones, which gets wrapped and wrapped and wrapped and wrapped and wrapped. But again, we'll be talking more about that in the future. But introduction, DNA gets wrapped up by proteins called histones into chromatin, which makes up a chromosome. So back to the physical structure of that nucleus, you've got a whole different bunch of points here that you need to be able to recognize. You've got overall the nucleus. There's a particular part of the nucleus called the nucleolus, which is very, very similar to the nucleoid. I'm dyslexic. These words all look almost identical. It takes extreme thought for me to make sure I'm saying the right one, and that's just true. No, these words all look very, very similar, and you need to be paying attention to if it's nucleus, nucleolus, or nucleoid. The nucleus is the whole thing. The nucleolus is where RNA and ribosomal RNA is made. So I'll talk more about that later. The other things to note here are you've got all these holes. So those are the are the pore complex made up of proteins. Okay, so you see we have a bunch of different pictures here, but what these are just focusing on is something as simple as what looks like just a little hole to let things coming in and out. We're going to find out in a moment, or we found out briefly, that's RNA takes that information from the DNA out into the cytosol. Well, how does it get from the nucleus to the cytosol? It goes through the nuclear pore. And you can think about it right now at this level, it's just a hole that things can come into and out of. But like you're going to learn in this class and in your future biology classes, everything, of course, is actually much more complicated than what we first tell you. So the nuclear pore is made up of hundreds of different proteins, and it's actually a highly regulated process. And this is really important and for some defense mechanisms to keep viruses from or certain viruses from being able to replicate. So everything is actually more complex than it seems. But Bio 181, we won't worry about that as much. But we will have to know about ribosomes. So ribosomes are the site of protein synthesis. This is literally where proteins are made. We're going to again have a whole unit on this. So you don't need to know the specifics. Ribosomal information stored in RNA that was made from DNA gets translated into making amino acid chains and proteins. So that's where ribosomes, that's what they are doing. Ribosomes can be in different places. They can be free in the cytosol. I think I've got a picture of that. They can be free in the cytosol, or they can be attached to what we're going to learn about in a second, the rough ER. Ribosomes have two different components. Ribosomes exist in prokaryotes and eukaryotes, but they're different size subunits. So they're actually different in between prokaryotes and eukaryotes, but they both have ribosomes. So I mentioned in a second ago about the ER, or the endoplasmic reticulum. It's a part of this whole structure that includes that nuclear envelope that's all continuous, which means it's made up of a membrane that flows through these. So some of these are separate, but the nuclear envelope is literally connected to the endoplasmic reticulum. And we're going to see through various vesicles transporting the plasma membrane. Okay, so we're now up to the endomembrane system. So this is a whole system that's made up of a bilipid membrane, so with that phospholipids, and it's going to include basically everything that's got these membranes that are moving back between each other. So that includes the nuclear envelope, which is connected to the endoplasmic reticulum, which then the membranes through various vesicles go to the Golgi, and it's lysosomes that are also made up of these membranes and vacuoles, and then ultimately, of course, the plasma membrane. So some of these are contiguous or continuous, and then others, it's the vesicles that are moving the plasma membrane. Okay, so we're up to the ER finally. I've mentioned it now many times. The ER is the endoplasmic reticulum. This again is continuous with the nuclear envelope, and there's two different parts. There's the smooth ER, which we're going to see in a moment, is smooth because it looks smooth and it doesn't have ribosomes, or the rough ER has ribosomes. So what does that mean? Again, it's continuous with the nuclear envelope. It goes right where somewhere we're going to be able to see that where it's moving, right there, right there. And you can see that this is rough because it literally looks like it's speckled with all these dots, whereas the smooth ER does not contain ribosomes. Now we know ribosomes are the site of protein synthesis, so the function of the rough ER is going to be largely producing those proteins, but also doing making some membranes, so those phospholipids. Um, the smooth ER, of course, is smooth because it does not have ribosomes, so it is not making proteins, but it's really focusing on all those lipids. So it's making new phospholipids, so new membranes and steroids. There's also some detoxifying effects where it's breaking down different molecules. So what is going to move then that membrane from the ER to the next, which we're going to see is the Golgi? That's vesicles, which literally bud off from the ER. So that you can think of as like a car traveling. So where is that car traveling? It's going to be taking proteins in particular, but also the membrane from the rough ER or the smooth ER and bringing them to the Golgi. So we're going to see in a second that there's a flow from the nuclear envelope. The information goes to the rough ER, and then those proteins go to the Golgi. There's actually different sides of the Golgi, so there's directionality to this. But what is happening in the Golgi? They are modifying what was made in the ER. So primarily proteins. It's going to be adding sugars and different sort of signals to say where the different components need to go. And then they're going to be shipped off in more vesicles. Okay, so we have different vesicles that are moving from these different components. And one of those membranous sac looking things is actually got a specific function, so it's called the lysosome. So again, what it is it's membrane bound, and it's got particular enzymes in it which serve to break down other molecules. So the lysosome has a particularly low pH, meaning it's got a lot of hydrogen ions. And so you can see here, it's going to fuse with other vesicles and ultimately break them down to reuse those macromolecules. So you can watch this own video. Another type of a large membranous sac is a vacuole. And there's two different types of vacuoles. There's a few different types of vacuoles, I should say. There's contractile vacuoles, which you can see here, where it's literally something that's going to be moving, opening and closing to contract. And you can see there, it just moved right there. So these can be in eukaryotic cells. This is an example of a protist. But the most famous vacuole is actually the central vacuole, which is only in plant cells. Animal cells do not have a central vacuole. They can have vacuoles, but not the central vacuole, which serves as the major storage of water within a plant cell. So the central vacuole can expand if there's a lot of water, and it can push out, ultimately up to where there's that cell wall, or it can shrink if it's very small if there's no water. So the endomembrane system, as a review, again, you want to be asking yourself, where are things going, and can I recognize what the different components are? So I've got a whole bunch of different pictures and a bunch of different videos that you can watch. Okay, so we're up to mitochondria and chloroplasts. And these guys are both important for power. And we're going to have whole weeks where we go specifically through the process of photosynthesis and aerobic respiration. So chloroplasts, of course, are the site of photosynthesis. So that's where a plant takes light, carbon dioxide, and water and converts it into sugar and produces oxygen as a result. Again, whole week on that. So we're going to see later on, there's this similarity of opposites between photosynthesis and respiration, where the reactants of photosynthesis are the products of respiration. The products of photosynthesis are the reactants in respiration. So it's water and carbon dioxide and sugar and oxygen. But the whole point of this is that we are making energy. So the point of photosynthesis is to make sugar. The point of aerobic respiration is to get energy and make ATP. So the mitochondria is the site of aerobic respiration. So this is how we take food, we eat it, we break it down, and we get energy. You're going to see that there's some similarities between chloroplasts, mitochondria, and prokaryotes. Why do they kind of look similar? If you look in the right ways, they've got these two membranes. They've got their own DNA. It's because these used to be free living prokaryotes. So this picture, a little bit more in depth on the chloroplast, but this is what I was saying about the origin of the mitochondria and the origin of the chloroplast. We now understand through the endosymbiotic theory that you had an ancestor to a eukaryotic cell that basically ate or engulfed a free-living, non-photosynthetic prokaryote, so something that was living on its own, eating, so to speak, sugars and other components, other macromolecules that were around, and breaking them down for energy. That got engulfed by an early ancestor, non-photosynthetic eukaryotic cell, and instead of breaking it down, the lysosome cutting it all up, then ended up benefiting that prokaryote. Now it lives in this nice, safe environment, and that host cell is like, boom, all of a sudden I got this thing that's just making energy for me. So it was a symbiotic, meaning benefited both cells. Ultimately, the prokaryote stopped being its own individual cell and became a part of that, what was now eukaryotic cell. And at least one other time, the similar event occurred where you have the engulfing of a prokaryotic organism, in this case, one that was doing photosynthesis, and it ended up benefiting both cells, and it didn't break down that chloroplast. And again, evolution, hundreds of thousands of years, that is now going to be one photosynthetic eukaryotic cell. So what was the evidence? Again, I mentioned this. It's their similar DNA. So both mitochondria and chloroplasts have their own DNA, and we actually can look to see in the sequence what that DNA is most similar to. They have different structured ribosomes, again, similar to prokaryotic ribosomes, and they can actually reproduce on their own. So there's some things we're going to look at for DNA throughout humanity, and you can actually look at the mitochondrial DNA as representing your mother. This is one of those great stories, or terrible stories, depending on how you think about it, where someone puts out an idea and at first the rest of science says, no way, and sort of laughs them off. And then through lots of hard work and continuing research to prove their point, they get proven correct. And it becomes widely accepted and that is the endosymbiotic theory by Lynn Margulis. And her sort of shout out to Darwin that she published was on the origin of mitosing cells to explain where these organelles came from. Okay, so next big topic though, the cytoskeleton. And at this level, you need to know that there's multiple types of elements of the cytoskeleton. What you can think of the cytoskeleton as is exactly that, the cytoskeleton. So it's the structure, just like your bones keep the structure of your body, that's what the cytoskeleton is. And so here you can actually see you've got green microtubules, so these are staining with different types they're staining different proteins, in this case microtubules or microfilaments. So the microfilaments are orange and you can see how they have some different structure there. I'm going to skip all of these those videos, but they're fun for you to watch. So one of the things that the microtubules are doing is they're helping things move. They are serving like the roads in that city, where you've got these vesicles that are going to and from different parts of the cell, moving proteins or moving waste, and they need to get carried. So using energy, they're going to be walking and moving these little feet-like things that they flop along this, in this case, microtubule. So you can watch some of these where you can see things coming and going. Again, there's different types of cytoskeleton elements. There's microtubules. Those are made up of tubulin. You can see that like some things aren't very creative. Microtubules, tubulin. Microfilaments, these guys are the actin, and these got those myosin motors that will move on them. And then you've got intermediate filaments. So microtubules are big, they're hollow tubes. Microfilaments are the smallest, and then the intermediate, shockingly, are intermediate size. So then the last thing we have is how cells can move. So cilia and flagella are the things that move. Flagella is what you picture when you picture a human sperm. It's a giant whip that's moving, that sort of flips and turns in order to project or to propel that cell forward. For humans, the only cell that we have that has a flagella, I shouldn't really say we, is in sperm. So males make sperm and they have cells with flagella. What we do have cells in all of our bodies have cilia. So cilias are just these little tiny hairs that sort of sweep things away. So on a protist, cilia can help the physical organism move all around in liquid. What we have in our cells, or our bodies, excuse me, are cells that are fixed, but the cilia itself is sweeping things away from it. So instead of the cell moving, it's all the stuff that's around the cell. And this is what's great for our lungs. It's keeping all that gunk out of our lungs. And when you like hock a loogie, what you're doing is you're getting all that debris that the cilia has swept up through your respiratory system. So you can watch some of these fantastic videos, along with a link to some more recent data on how sperm are actually moving. And then the last thing are just talking about the cell structures, the outermost cell structures, and actually it's not really the last thing, but we've got a cell wall that's in plants, and this cell wall we're going to find out in the next unit is really important for keeping that hard structure. So we're going to see that animal cells can shrink, but plant cells have this hard structure just like if you picture a wall. So I want you to think of the cell wall like you think of a chain link fence. There's definitely holes where things can come in and out, but there's a hard place that you can't pass. Animal cells, we don't have cell walls, but what we do have is a whole bunch of stuff outside of our plasma membrane. So here we have the cell, we have inside the cell in the cytoplasm. Here we have the extracellular matrix. So the extracellular matrix is full up of lots of proteins, some sugars, other stuff that's signaling between different cells. And so it's called the extracellular matrix, and it's got primarily collagen, which is if you like pinch your cheek right now, that's mostly what you're feeling. There's all that protein, which when you get old starts to break down, and then you get wrinkles. Okay, so again, what do you need to know about this chapter? You need to know, even more than listed here, the structures and their functions, and then what types of cells they are in. So can you put all of these different structures and organelles into a Venn diagram where you have here, you have all eukaryotic cells. So here you have the circle for plants. Here you have, it's actually a rectangle for animals. And here you have a rectangle for prokaryotes. So things that are just animals and plants, you put here. Things that are all of those different forms of life here. Things that are just prokaryotes here. Things that are plants and bacteria here, etc. So you should be able to then fill out and know where all these different structures are. Again, you need to know where it is, be able to recognize it, and know the function. Good luck.